Today you're listening to PSGR. My name's Jodie Brunning, and today I'm here with Andrew McCabe, Tessa Hiscox, and Professor Jack Heinemann. And together with Brigitta Kurenbach and Sophie Walker, they published a submission by the Centre for Integrated Research in Biosafety, INBI, from the University of Canterbury here in New Zealand. And their 38-page INBI submission to the Parliament Health Committee on the Gene Technology Bill 2024 can be accessed via Professor Heinemann's profile. All the authors of this paper have previously authored scientific papers in the field, so they have definite expertise connected to our discussion today. Gene editing technology directs the location of change in a genome, and this happens whether you add foreign DNA or DNA from that organism and take it out again. And all gene technologies increase the mutation rate by changing the frequency of mutations above background levels. Now, the Ministry for Business, Innovation and Employment, and Judith Collins, who was the minister when this bill was constructed, right out, they exempt a powerful mutagen. Gene editors, the technology, are themselves powerful mutagens. Now, powerful mutagens by convention have strong oversight and control. In mammals, for example, they can cause cancer. Your submission explains that MB are focusing on an outcome, a gene-edited organism, but they explain why it is essential that New Zealand includes the tool in risk assessment. Now, New Zealand would end up having, as you say in your paper, the most extreme combination in the world of proposed species breadth, microorganisms, plants and animals, and process, e.g. STN2 exemptions. Australia is also similar in species breadth, but not in process exemptions. Now, MB believe that processes of site-directed nuclease activity, so the SDN2 that we see here, that has a nucleic acid template, and oligonucleotide mutagenesis, that's ODM, are completely distinguishable from conventional breeding. Now, MB would exempt these processes, but MB's bill prevents the regulator from conducting risk evaluations on these particular processes. The regulator's powers throughout the bill restrict then how much that regulator can understand risk and mitigate it. Now, only a few, relatively small number of plants have been bred for use as crops and industrial feedstock, and animals for livestock or feedstock. And fungi and bacteria are used for mainly secondary process products. Now, using site-directed nucleases, SDNs, most of the species that would be modified intentionally using these mutagens would effectively never be modified by a similar process through conventional breeding or in nature. Now, I've got this, I've drawn what I'm saying here from INBI's submission. Now, Jack, in your submission, you state the processes capable of creating similar hazards do not necessarily create similar risks. Creating hazards indistinguishable from those which may be created by conventional breeding is not the same as creating risks indistinguishable from conventional breeding. Please, can you explain this, or one of your colleagues here today, using the hornet's nest example in the paper, and we will put the image up for members of parliament, officials and public to see.
Yeah, happy to do so. The point is, and this point was also made really effectively by the US National Academies of Science, when two things each have the same ability to hurt you, where you are in relation to those things matters. So it might be that a car can cause the same damage to you as a meteorite. But meteorites are very rare, and they don't purposely drive on roads where you also find people. You can't exclude the exposure potential from a risk assessment. There are many different ways in which the products of a gene technology may have very different contexts than those that are produced by conventional breeding. An example that makes them very different is they attract different intellectual property rights instruments and therefore are also tailored to very different specific uses and therefore create different contexts just from that pathway. But also because at its root, a biotechnology is an ability for people to make natural things happen at unnatural rates and at unnatural scales, geographical scales. Because of that, we also will artificially create in contextual exposures that you wouldn't by conventional breeding. For example, I saw a trail produced by defense analysts talking about using these kinds of gene technologies to make bespoke changes to organisms on the battlefield. That you can't do by conventional breeding. There's just not a time or species level ability to do that. But there is by gene technology, even if in 500 million years or so, the same thing could arise by conventional breeding.
Okay. Now, New Zealand would have the most extreme combination in the world of proposed species breadth: microorganisms, plants, animals, and processes, e.g. the SDN2 exemptions. Now, I'm repeating this to help us understand. MB would lock in exempt processes as non-notifiable. MB and Judith Collins have not only undertaken economic or risk benefit assessment, they haven't assessed how many organisms would then be exempt and non-notifiable as a proportion of all the GE organisms that could potentially be developed for release in New Zealand. Can you estimate what proportion of all GE organisms would be exempt and non-notifiable? And if you can't, can you recommend a scientist in New Zealand that has evaluated this and could understand this?
Well, we're not aware of anyone who's evaluated this. The point is that exempt activities, which is a weird way of saying particular products, and non-notifiable activities, which is a weird way of saying the process by which you make these things, would be outside of regulatory overview. And that means that you don't control what organisms are exposed to the genome editors, and you don't control who exposes them to the genome editors. You can use extremely common and areas in your home or around a school or on your lawn. None of these would be regulated, and by people who aren't technical experts in the field. And I don't say that to be dismissive, because technical experts can do pretty stupid things too. But at least under current regulations, even if a technical expert does something stupid, it's inside of a contained facility. And all the stupid things can be rendered inert and not released inadvertently, hopefully, into the environment. That would change. So the proportion of organisms that are altered using these exempt processes would be astronomical. To give you some sense, if I do this outside, I'll be exposing bacteria. There are billions of bacteria per gram of soil. There are hundreds of millions of bacteria per cubic meter of air. All of these would be simultaneously exposed. And it's a fallacy to think that we know the genomes of all these organisms, and that the tools that we use to direct those mutagens to particular DNA sequences in their genome, that those tools will exclude sequences that are in the genomes of all those billions of different species. Our databases, even if they could tell us these things by being complete, are not complete. On top of that, all of our cells have latent viruses, and some of our cells may be under an active viral load from an infection, even if it doesn't result in symptoms. Those viruses comprise a virome for which we have almost no database information at all. And that means inadvertently, any of these tools used at any time could be making changes in organisms that we have no intention of changing. The vast majority of those changes will probably be irrelevant from a biosafety point of view, but at the scale we're doing it and the failure to record what organisms we're doing it in, and the inability to contain them and destroy them before they're in the environment, that combination of new variables we cannot dismiss as negligible risk.
And there is in the scientific record, there is a record of inadvertent alteration of not just the DNA they thought they were working on, but DNA that was next to the cell, for example.
Well, and I would just point out that even in the intended organism, you can't control all the unintended changes. The use of the word precision by practitioners is really misleading in my view. These reactions are highly efficient, but we do not completely control the location in the genome at which they are being efficient. But even beyond that, there's a problem, and that is the reagents we use, the specific ingredients in the reactions. These come from biological sources. And each of those biological sources is contaminated with DNA from the pipeline of production. So even if you're intending to only use a type of process that creates an exempt activity, an activity where the product has only had a site-directed nuclease damage the DNA, and that damage should have been repaired using processes that are endogenous to the cell, and therefore not producing a new sequence from another species, for example. Even if that's your intention, you cannot rely on that being the outcome, because there's contaminating DNA in your reagent line and potentially just from the contamination of where you're doing it. And if it's not in a contained laboratory, there's even more contaminating DNA sources. So amongst the mix of products that you invariably make every time you use a gene technology will be a substantial number of those that we traditionally call transgenic organisms, and which this bill continues to regulate in higher risk tiers. The exempt activities cannot be considered or assumed to be exempt activities until they're verified and proven to meet the exemption criteria.
That does make sense. Can one of you please explain why there would end up being more GMOs in New Zealand, including being released into the environment and potentially in our food because of this bill by INBI?
I think that's still me, is it?
It's up to you.
Yeah, okay. Well, once again, there will be more failed GMOs in the environment. That is, ones that people didn't intend to make, ones that didn't work out the way they intended, but for which they don't even know they're making them, because you can do this in uncontained facilities, and those exposures then can't be destroyed. The results of those exposures can't be destroyed prior to entry into the environment. Now, we won't have any idea when this happens, how often it happens, and in the diversity of species in which it happens. And that means that as our detection techniques improve, because that will happen, it is already happening, we can't assume that we will be as blind to these changes in the future as we might be now. As that happens, then these unknown organisms, which will be regulated in other countries, can contaminate our exported goods and cause trade disruptions. Countries such as Brazil and all of the EU countries have what's called zero tolerance. They will allow a certain amount of admixture, a little bit of contamination of genetically modified organism, provided that it falls below a certain quantitative threshold, and it's a known genetically modified organism that has benefited from a risk assessment. If it is an unknown genetically modified organism, there is no tolerance in the entire a consignment is condemned.
Can one of you discuss the recent United States decision and where the European Commission stands, and are their regulations the same as New Zealand?
Yep, I can address that. So the US is normally seen as one of the most liberal countries in the world in regard to their genome editing regulations, but recently they've removed the phrase indistinguishable from conventional breeding from their legal requirements, which is something that obviously Jack has talked about before. This was done because this phrase doesn't actually imply that conventional breeding makes an organism of acceptable risk. So making harmful organisms in another way is still not okay. Even if it is similar, you're still going to have that same problem. The National Academy of Science in the US, who were the ones that were in charge of all of this, also said that you can't determine risk from equivalent hazards. So that exposure, as Jack has talked about previously, is still a critical part of these risk assessments that need to be performed. In regards to the EU, they're still only discussing changes. Nothing has actually happened yet, and they may not change at all. Even if they do, this is still only going to be involved with plants, like a lot of other countries, with microbes and animals not being brought into this discussion. Along with that, in the EU, whilst the EU themselves may have one ruling, the countries within the European Union still have to get on board with this, which may or may not still happen. So this will see New Zealand as far more extreme in all ways than either of these two areas.
No evaluation has been undertaken of the extent of deregulation in our key export markets, which may also differ by industry sector, say kiwi fruit or dairy. Have MB or MFAT undertaken this evaluation?
I wouldn't know for sure, although if you look at the regulatory impact statements from these different regulators, they're pretty vague on that, and sometimes they say they anticipate significant problems, which should be canvassed prior to adoption of the law. The Food Standards Australia New Zealand has done a global look at asynchronous changes in regulations around the world. So they've put together, and we reproduced this in our submission, they put together a table that shows all the countries who have made changes and all the countries who are talking about making changes, which this government has conveniently lumped into one number to suggest that lots of countries have changed, which isn't quite right, because anybody who's only talking about it may not change. But when you drill down more deeply, there's lots of differences around the world in what changes are both being discussed or have been made. Most countries, there's only 2 countries in the world that haven't required a verification step. That is, that you prove that the organism you want to put in the environment is the organism you said you made prior to release into the environment. Only 2 countries, and those are Australia and Canada. But Canada only allows this for plants, so that's already different. Whereas Australia allows it for all kinds of organisms, it doesn't allow it for all kinds of processes, as you mentioned in your introduction, that New Zealand would. So we would leapfrog Australia as the most radically deregulated country in the world. Even the UK, though, who has left the EU, have made changes only to plants and animals, not to microorganisms. So they have taken a step back even from Australia's extreme position.
Thank you. Now, can you please, or one of you please explain why you explicitly state that all gene technologies can only be used in containment facilities, and why is this such an important recommendation?
Sure, I can do that one. So as we've touched on earlier, a lot of gene technologies are based on template sequences, which guides where the gene technology binds and does its activity. So the sequence is made to match the gene that we want to change, but DNA can also squeeze into this template sequence if it's close enough. So if we think of putting on a pair of pants that's too small, um, we can squeeze ourselves into the pair of pants, and at the end of the day, we're still wearing pants, even if they're not made to fit us. So it's the same for DNA. If the match is close enough, it can still bind and create that change in the genome. So that's an unintended change from gene technology, and that can happen in the organism that we're interested in. And the reason we're recommending doing it in a containment facility is that we can contain all the unintended close enough changes that have happened inside of that containment facility within that organism that we're interested in. But also for a containment facility prevents the exposure of other types of organisms that may be exposed. So if we were applying a gene technology to a field of plants, but in that field of plants, there's also a range of other organisms, such as earthworms or pollinators, microbes in the soil, or people working in the field. And when we've applied this change, and it leads to all these unintended changes in those genomes as well, it means that those changes are automatically, immediately in the environment where they can spread and be passed on. So the cat is out of the bag at that stage and the damage has been done. So we think a containment facility requirement would alleviate that.
Though it might be technically covered by other legislation, such as the Biosecurity Act, would make it easier for unscrupulous players to cause economic harm to New Zealand. As an example, we are heavily reliant on dairy as a primary export. And dairy cows are heavily reliant on eating grass and stuff. And that grass and stuff is reliant on earthworms, who make sure that the nutrients and soil conditions are best suited to those pasture grasses. Very, very small changes in the productivity of an earthworm magnify up to the productivity of a dairy cow. And if you were to use a genome editor simply as a mutagen, sprayed on cropland, on pasture land, with the specific intention of undermining the viability or activity of earthworms, you could have a major impact on our production capacity. That would all be technically legal using exempt activities. And if I were a dairy company in a different country, and I even just wanted to see whether spraying these on earthworms could be a good thing, I would do it here, because if I was wrong, it's not my country that will suffer.
So it's the lowest entry point, the lowest bar. That's what we would become.
Mm-hmm.
Thanks for that. Food for thought. I do hope the big industry sectors will be listening to this interview. And a final question, can one of you or all of you discuss why you think retaining the Hazardous Substances and New Organisms Act 1996, the HAZNO Act, as the most practical solution instead of the gene technology bill, which would become the Act, and why risk tiering inside the HAZNO Act instead of risk tiering outside that prospective Gene Technology Act might be the best solution for New Zealand?
Yeah, I can take that. The difference between the Gene Technology Act that's being proposed, the bill that's being proposed, and HAZNO is not the range of things you can do. HAZNO allows you to do everything already that enable the use of a gene technology. There's a few more hoops, a few more obligations, a little bit more accountability that tracks with your use under HAZNO, but it doesn't stop you. You just have to show that your product is safe and that there's a reasonable basis for wanting to put it in the environment. And sure, any regulatory process has its costs. The question is whether or not they're proportionate and appropriate. What this act does, different from has-no, is it says, or it hopes, that suddenly there will be a new utopia of gene technology outcomes simply because nobody has to tell you what they did. And because they don't have to tell you what they did, that is going to massively reduce costs. Okay, well, personally, I'm not convinced of that. If I'm really trying to make something for a commercial purpose, I'm probably going to have pretty much the same costs anyway, because I'm going to want to control my intellectual property. I don't want that organism to be captured by anybody else during the process of production. And for all sorts of reasons, I'll be using a containment facility just for that. And the collateral will be that my organism is recorded. Now, so I don't think this is going to reduce that many costs. And it opens us up to those risks that we've discussed earlier in this interview. If you do believe, though, that has-no is too restrictive, then the part that you could tinker with is the transition from the lab to the environment, rather than removing any pre-commercial or research-based development, removing that from some kind of use in a laboratory. If at the point in time, and this is what other countries, I mean, India explicitly says this, they restrict it to plants, but if you do it in containment and you can show that the plant you made is the one you say you made, then it's expedited for release. It meets the exemption criteria if the description conforms to an organism at low or very low risk. And that's predetermined exemption criteria. Then you don't have to go through a lot more process to go to the environment. So they have achieved a tiering that is highly compatible with our has-no act, where you would continue to use containment in pre-commercial or just research use. And when you want to release it, provided that it falls within some kind of defined tier or license, then you can release it. But that verification step is essential. We already know how many times somebody has said, this organism is exactly what we're telling you it is, and later it was found to not be. So we have to have reasonable standards of verification. We have to have processes that provide confidence that whoever is declaring this organism as being what they say it is can be tested and challenged and shown to be true, and then release it. And that seems to me to be the definition of proportional.
Well, that makes absolute sense. And just a final question, because I always do this. When the regulator in the current bill doesn't seem to have any obligation to undertake monitoring, so that when an organism is released, there may be study of this and actually the resources to study that. Do you believe that should be in any legislation looking at GE organisms?
The EU requires monitoring under their present, but only in the EU. And there's a good reason not to have monitoring in the gene technology bill before us. You don't know what you're making, so you can't monitor it. You have to know what you're making in order to have the tools to monitor it. So if you're exposing millions of species simultaneously, you won't know which one to look for, and which ones, which of the many that you have to look for. So it would make no sense to have a monitoring provision for something that has no other regulatory oversight. However, if through the simple solution of requiring all research and pre-commercial development to be in a registered containment facility, you will know what that organism is. And if you wanted to monitor it, you would have the tools to do so.
That makes absolute sense. Now, do any of you have any final comments or thoughts that might be useful for anyone watching this? It's a Friday. Have a beautiful day. Thank you so much for coming on at late notice. We're PSG are very grateful, and you have a lovely weekend.
Thank you.
You.
Thank you very much.
Thank you.